Okay, hi everyone. Welcome back. Happy Monday. And yes, I just uploaded the slides for today. I know I'm kind of reformulating things. So while you're downloading the slides, let me explain. So if you look at the syllabus or actually let me just pull it up. Let's look at the syllabus. And yeah, if we pull up the syllabus, you might notice okay, after chapter 12, things get different. And they do get different. And notice that, you could use actually the OSU chapter 12 and the OpenStax chapter 12 are pretty similar. But chapter 13 onward is where they really start to diverge, especially from the Martini book. So the thing about the nervous system is incredibly complex. And I don't think there's an easy way to teach the nervous system in a linear fashion. So that's one of the hard parts about teaching the nervous system. Now, let me explain things before we start. Here's an interesting question. And it deals with the nervous system, obviously, but why shouldn't you trim your cat's whiskers? Now, think of that while you're downloading the slides and pulling them up. Okay. So here's explaining more about this. So the Oregon State University AMP, it is free online. There are links to that in the syllabus as well. And you can just search for Oregon State University Anatomy and Physiology. So don't worry about that. Oh, you try to open it, but it says error in the document. Let's see. I don't know. It's opening okay on my end. I'll try to upload it again later, but yeah. But okay, so back to Oregon State University's Anatomy and Physiology. So it has all the same information as everything else, but the thing is that, so actually in previous years, before maybe last year, we started using Martini's Fundamentals of Anatomy and Physiology, which has more detail and I think better prose overall. Now, OpenStax, what they did is basically, this is the way I think they tried to do their current version of the nervous system. Somebody, I'm not sure who exactly on the authors, they really love the neurological exam and probably neuroscience, and they really wanted a chapter on that. And basically, they took everything else, chopped up, and tried to make a whole chapter, maybe like a capstone sort of lecture or chapter for their book. But then this kind of made the other chapters very uneven. So especially the autonomic nervous system, this part kind of irritates me because some of that, the autonomic nervous system standard curriculum is now kind of distributed between the two. And if you look at especially chapter 14 in OpenStax, I think is the most uneven chapter. Sometimes there's only a paragraph, sometimes there's a lot of stuff. So this is why I think OpenStax is a little weird. Yeah, so I think that's why I think this is where, for the nervous system, I suggest using my lectures first. And I apologize if this one's a little crunchy because this is my first time teaching off the OSU version. But I think the OSU version is the free version that corresponds more and is more organized. Because even the Martini one, I gotta admit that chapter 15 is a little weak. It's chapter 15 for Martini is almost like everything in the other chapters we didn't have room for. It's my least favorite chapter in the Martini book, but it's this is important information, but it's kind of like the leftovers. Now, this is another thing. I know I did some sleuthing, and with the instructor resources for the OpenStax version of Anatomy and Physiology, look what chapter 14 was titled before. It used to be like the Martini version where it was the brain and cranial nerves. And this is the current chapter 14, which is kind of like a very bare bones leftovers of whatever they tried to dice among the other, and slice and dice and distribute among the other ones. This is the thing with the OpenStax Anatomy Physiology. It seems like chapter 16 is their baby for the nervous system, and all the other chapters except chapter 12 kind of got the leftovers. So, yeah, this is what we have here. So basically, this is what I think happened with Martini, and this is why the nervous system is kind of jumbled, because it's kind of hard to teach the peripheral nervous system while pretending that the brain and spinal cord don't exist. But at the same time, it's hard to just like say, okay, this is sensory, but let's ignore motor altogether. I think that's why there's no real easy way to teach the nervous system. So bear with me, but I think the OSU makes more sense to me. So yeah, OpenStax, I think they took the concepts from Martini's, sliced it up, and OSU tried to stitch it back in a more logical fashion. And this is how Martini looks compared to the OSU. So I think the OSU, it does shift things some around from the Martini version, but I think it's a little more logical. And for the Martini, okay, so this is another thing too. You might notice that I put the autonomic nervous system earlier in the semester. I don't save it for last, and there's a reason behind that. The autonomic nervous system is where we get really into like the molecular biology and like a lot on the cellular and tissue levels and pharmacological levels. And that's a harder concept. Special senses are they're fun. One, I put them at the end because I think they're easier than the autonomic nervous system, and I think they're fun. So I like to put the heavier stuff up a little earlier so you have more time to study that and leave the fun special senses for the end, because yeah, the special senses are part of your somatic nerve, or just like your sensory pathways, but I'll put them like the Martini version. I'll put them at the very end of the semester. And this is the thing, you might be like, okay, so we're getting the OSU, but are we missing out on anything? And OSU has all the information from OpenStax. You can compare the chapters, especially the chapters that correspond directly. OSU used OpenStax as a basis. So OpenStax is an open educational resource. So they took that information, they just reorganized it in a more logical fashion than the current OpenStax version. So this is why I'm switching to OSU for the rest of the semester. You can still get this information. Maybe you do like the OpenStax version. Maybe you do like their current layout. If you read it from the chapter end to end, you'll get the same information from the OSU version. So again, don't feel like if you use one or the other, you're missing out. But I'm just letting you know that from this point on, I'm using the OSU version more as a schedule because I think it makes more sense and it's more logical than the OpenStax version. Yep, and let's see, since this week, again, this is an adapted version of OpenStax AMP, so you are getting the information, you're not missing out on anything. Week 12, yeah, if you did all the pack-back question, if you got 100% on, or not 100%, but if you did your one question, 4 responses, they all met the minimum CP, then you should be done. If you have 1,200 points, you're done. Nothing, you can still participate, but I mean, you're capped at 1,200 points. So that's what we have for pack-back. Okay, so a quick review. Remember that your central nervous system is what's the components of your central nervous system? You have your brain and your spinal cord, right? And really, that's it. It's just your brain and spinal cord. Your CNS is your brain and spinal cord, and yeah, but everything else that branches off from the central nervous system is considered your peripheral nervous system. And I don't know why they're not showing cranial nerves here, but pretty much everything else that's not your brain and spinal cord. So all your other nerves, your cranial nerves, your spinal nerves, these are the branches from that central nervous system to the rest of the body. And that's what we're going to be covering for the next few lectures. Now what we have here is so what we have is like, okay, stimulus. So some sort of signal from somewhere in either your external or internal environment. And a stimulus is going to activate something called a receptor. So we have our sensory receptor neurons. And sometimes you might hear the term sensory receptors or just receptor neurons. When I tend to say all three, I tend to say sensory receptor neurons, so you're not going to confuse them with other use of the word receptors. So what happens is that stimulus, some sort of signal, some sort of change, causes these neurons to depolarize. And remember, this is everything with our previous 2 lectures regarding action potentials and ion movement and neurotransmitter release and different types of synapses. So depolarization results in action potentials, and neurons pass action potentials to other neurons and other cells. So these action potentials get relayed from neurons to other cells, and then they eventually, and this is what we call pathways. So pathways, and here we have polysynaptic. Why? Because these neurons are extending their axons and synapsing with other neurons that are relaying the signals, and eventually it reaches a central location we call the central nervous system. So at least all signals that are incoming to your body are relayed somehow to your, at least your spinal cord. Sometimes they're not relayed all the way to the brain, but they do reach your CNS, and this is again why it's a little tricky to teach the peripheral nervous system and pretend the CNS doesn't exist. So yeah, but the way I get around it is like, okay, I'm going to talk more detail about the sensor or the peripheral nervous system and leave the details of the CNS when we get to it. This week is the last week we have to do pack back to receive full credit. Yes, so if you have, I think I have updated until week 10, but yeah, if you have 1,100 points after the previous or this past Sunday, then you only have 1 week left. Okay, so this is where it can get a little tricky, and especially the Martini book and lectures, and heck, even I sometimes use this interchangeably. But receptor, so it's the same word but different use. It's just about scale. So when we're talking about sensory receptors, are we talking about the cell level? Are we talking about a protein on the cell? Or are we talking about a small structure on the cell? So sensory receptors or neurons, these are cells, and I think this is why I often sometimes just say for clarity's sake, I say sensory neuron or sensory receptor neuron. So I'm trying to say that is a cell I'm talking about that detects something. Now, you also have cellular receptors, which are proteins on cells. So these are small molecules on your cells, but they detect some sort of change in the cellular environment. And sometimes, especially if you talk to a molecular biologist, or especially when we get to the endocrine system, when we talk about hormones next semester, often when I use that term receptor, I'm referring to some sort of protein that detects some hormone. So I might say protein receptors or cell receptors when I'm talking about a protein on the cell. So again, they both detect something, but is it on an overall cell scale or a smaller protein scale and molecule scale? But the thing is that they both detect environmental changes and stimuli. So if you're getting confused, try to think like, okay, are they talking about a cell here, or are they talking about a protein here? So again, so most of the time, not all the time, but most of the time when I talk about a sensory neuron, when I'm talking about sensory neuron, I'm talking about cell. But when I say sensory receptor, and most of the time in the textbooks when they talk about sensory receptors, they're talking about neurons. They're talking about cells. Okay, back to elementary school. What are your 5 senses? And I'm pretty sure they still teach it to some extent in elementary school. Well, the 5 senses you probably heard are like vision, sight, hearing, smell, taste, and touch. And just like how physiologists and anatomists have to make everything more complicated and have more complicated terms for common terms, we also have these more medical terms for all these. So vision, you probably heard, like vision, audition. And then olfaction, you might have heard, but that's the more medical term for your sense of smell. Gustation with taste, and somatosensation. I know, why not just call it touch? But actually, somatosensation is more than just touch, but it's talking about like, okay, sensations throughout the body. Yeah, so soma refers to the body, and then sensation is sensation. But the thing is that are there just 5 senses? And there are more senses. There includes like proprioception, pain, temperature, pressure, vibration, and touch is not only just one sense. It can actually be broken down into further other senses as well. And this is things like fine touch, crude touch, light touch. So fine touch is like, okay, you can tell the details of something you're touching. Crude, just you know where something's poking you or touching you, but you're not exactly sure or able to like, what shape is it or where is it exactly? Like if a fly or like an insect lands on your face, you definitely know where it is and you know where to swat. But say like a fly lands on your back, you probably know it's like lands, it's there, or if you even feel it, you know it's there, but do you know exactly where to swat? So some places on your body are more sensitive than others, so the fine and crude touch, that's part of that. But like pain, temperature, like temperature, is that or like things like proprioception, that's not exactly a sense of touch. And I mean, you could say like these are related to touch, but they're more about physical contact, but these are more discrete like sensation and stimuli. Now, another term you might hear are, instead of sensations, are modalities. And I know this is another term where people are trying to make something simple even more complex. And so these are all, sometimes you hear that term sensations used interchangeably with modalities, which is just a way you perceive the world or perceive information about something. Now, just because we can't sense it doesn't mean that something isn't there. Like what's something in our environment that we can't detect? I mean, unless you're a bird or something, or one of those what was it, tetrachromats, you can't detect UV light with your eyes. Does that mean that, or you can't say like, okay, there's more UV light right now than there was before. So we can't detect UV light with our eyes unless you're very, very special or like an X-Men or something. But does that mean it's not there? No, we just don't have the modality and sense ability to detect that. Or things like magnetic fields. Some animals can detect magnetic fields, but can we detect magnetic fields? No, but because we can't detect magnetic fields with our sensations and some modalities, does that mean these magnetic fields aren't there? No, they're there. We just can't detect them. So this is why sometimes they call it modalities, because it's just like, okay, this is how we perceive the world. And yes, our experiences are very real to us, but it's also getting to the point that our sensations, we get a lot of information from everything, but we don't exactly get all the information from our environment. Okay, so back to senses. So let's talk about general versus special senses. So general senses and special senses, and this is what you should definitely know, is what's the difference between the two. Well, general senses these senses are not located just to specific parts of your body. These can be detected widespread, and these are able to be sensed through multiple parts of your body. Whereas special senses, they have specialized organs that are specialized in that sense. So general senses include proprioception, pain, temperature, pressure, vibration, touch. So all those ones I talked about, these are general senses. So can you feel pain when someone, like, say they flick your earlobe? Yeah. Can you feel pain when someone pinches your arm? How about when someone pinches your thigh? Or if you have shoes or stub your toe, you can feel pain at multiple parts of your body. Can you feel if something is cool on your head? Can you feel something is cool on your back? Can you feel if something is cool on your leg? Yeah, you can feel temperature at multiple parts of your body. So that's the general sense. Now, special senses, these are the ones that almost directly map to those 5 senses you probably learned as a child. So things like vision, like, yeah, we see it with our eyes, but can we see with our tongue? And I know somebody's going to say, what about people who have vision deficiencies and they can read Braille? Well, this is using touch. They're not actually detecting light like we do with our sense of vision. They're able to get information, but it's not actually taking photons and converting it to some sort of visual image. Now, hearing, can we hear with our elbows? Can we hear with our feet? You can detect vibrations, but can you actually detect convert it to some sort of sound or some sort of sensation of sound? No, not really, right? And how about your sense of smell? Can you smell with your eyes? Can you smell with your, I shouldn't say, well, you can, or actually it's interesting, your sense of olfaction is linked to your sense of taste, but they're two different sensations. I was going to say, can you smell with your tongue? Well, it does affect your sense of taste, but can you taste with your eyes? Oh, ouch, yeah. Can you taste with your ears? No, you can't. Can you taste with a random other part of your body? Not really. So again, special senses, they're located in special areas. There's equilibrium as well. So this is a sense of balance, and this is in your inner ear, something we call the vestibule. So this is what we have with the the special senses. So that's the interesting thing, is that special senses, we have the 5 special senses that are almost like the 5 senses you learn as a kid, but instead of touch is actually a general sense. So we're going to move touch there. And equilibrium, this is with your inner ear. So this is like, yeah, this is only located with that. Okay, so the thing about all these stimuli, so why do you have so many senses and why do you have so many sensory receptors? Well, thing is that, say it's cold outside, do you want the cold to always cause you pain regardless of whether it's warm, cold, or whatnot? So you don't want painful things like someone touching you to be painful every single time, and at the same time you don't want something like, say the change you move a joint and all of a sudden you feel hot. That would be kind of weird, right? So you don't want these senses to be mixed up with each other. So different sensory receptors detect different stimuli. So this is actually a very interesting one. So thermoreceptors, thermal sounds like heat, right? Like sounds like a thermos. Chemoreceptors detect chemicals, and osmoreceptors detect solute concentration, which affects osmolarity. Now this is an interesting term, nociceptor. So noci is kind of, think it like the same root word as noxious. It's something that causes harm. So these nociceptors, these detect painful stimuli. Whereas you have other things like tactile receptors, baroreceptors. Baro is actually very useful, like a barometer measures pressure. Same with a baroreceptor cell, that detects pressure. And proprioceptors detect more of the position of your body. And you might be like, wait, position of your body is a sense? And it's one of the senses, like, it's so natural to us, we kind of take it for granted. But I'll get to that hopefully by the end of the lecture today. But yeah, so proprioception is one of these things, general senses we like, wait, that's a sense, but when we lose it, it really starts to affect us. So yeah, basically for our body and our nervous system to detect it, you need some sort of receptor for whatever change in the environment you're trying to detect. And you also have specialized protein receptors for different types of sensor receptors. Ooh, dude, nociceptors make you itch too. I actually have to read up more on that, because like I think what the latest status of that debate is, like itch. There's like, is it somatosensation? Is it nociception, or is it its own sense? I know itch is actually kind of a very interesting sort of sensation, and I remember like I attended a lecture from somebody who actually specifically studies itch, because like the context she was studying it was like in the hospital, and it's like, say somebody has a persistent itch, but you can't really find out what's causing that itch, and that itch is affecting their quality of life, and where they're constantly distracted by an itch. Like, yeah, that's why she was researching it, because she was like saying like these patients have like a persistent itch that they can't get rid of, and it's totally affecting their life, and everything they do is constantly distracted by that itch. But hey, actually that sounds like a great tack-back question. Call dibs on that one. All right, that's a good one to end on. Okay, now let's talk about protein receptors. So again, just like sensory receptor, all stimuli need to be detected by that can be converted to one of our senses, needs some sort of cell. You also need some sort of thing in your cells that detect these senses. So things like thermoreceptors, they have ion channels that open in response to different temperatures. Chemoreceptors, they can detect chemicals that act as ligands. So again, ligands and receptors. Ligands are the keys, receptors are like the locks. So chemoreceptors can like detect different chemicals, and mechanoreceptors, these also are like, so mechanoreceptors rely a lot on mechanically gated ion channels. And let's give an example of how a protein can how a protein can tell a sensory neuron what to do. So here we have a mechanically gated ion channel, and say you have something like, remember that mechanically gated ion channels, they open in response to some sort of distortion and some sort of movement in the cell membrane. So pretend we have something that touches or distorts the membrane, and that causes this to stretch and open enough so that we have an influx of sodium going into the cell. Now, what happens to the cell's membrane potential if we have this open sodium ion channel? Well, it's going to it's going to slightly move the membrane potential of that cell toward the positive area, right? So once it goes towards the positive direction, now due to the positive charge of all the sodium ions, it's going to move this closer to the threshold. And once a neuron crosses this threshold, what's going to happen? An action potential is going to happen, right? And why is that? Well, because all these voltage-gated ion channels open in response to this increased positive charge inside the cell. So that sets off an action potential, and this carries the signal, so it can travel along the axon, set off a series of action potentials, and then carry the signal off to other cells. So again, this is how you go from protein to a cell signal to an overall sensation signals. Now, nociceptors, there's not one single protein that detects nociceptions. And why is that? Well, think of the different ways that pain can be caused. Like, for example, temperature. What if you touch something that's too hot, like a stove? Don't do that, by the way. Or if you touch something, or what if you hold on to a cold object for very long? Eventually you start to feel a very painful sensation, right? So these some of our nociceptor neurons, they can detect different extreme temperatures that cause pain. So too hot, too cold, some sort of uncomfortable temperature. So these temperature-gated ion channels, in this case I'm showing something that's causing pain in response to heat, but it can also be in response to cold as well. So these are going to open and cause these neurons that detect pain, these nociceptors, to depolarize. Or how about mechanically gated ion channels? What if something pierces your skin? Or what if you've ever been in elementary school and if you've had this where they grab your arm and they do this motion with your arm, it's super painful, right? And what's happening when they do that to your arm? Well, they're distorting all the tissues and thereby all the cells and the membranes of the cells in your arm. So that's going to set off a whole bunch of mechanically gated ion channels on nociceptors that detect nociceptors that are also mechanoreceptors that detect some sort of distortion and some sort of change in your stretch in your tissues. And then you also have ligand-gated ion channels. So you can't have chemical things like pepper spray. That's a chemical, that's capsaicin. And what's going to do is bind to these nociceptor ion channels, these ligand-gated ion channels, and it's going to depolarize these ions. So you can cause pain through different ways. Extreme temperatures, through extreme sort of mechanical stress, through chemical exposure. So there's no one singular nociceptor ion channels. It's just showing you that there are different ways of triggering these sensory cells, these sensory neurons, by having different types of proteins that detect specific changes and stimuli in your environment. Now let's go very quickly. So these are tactile receptors in the skin, so all the way back to the integument. Now, am I going to talk to extensively about all the strata again? Not necessarily, but now you're looking at the dermis here, and you have all these nerve endings. And this is why when you burn through all these layers and burn through these nerve endings, that's why when you reach that subcutaneous layer and have a 3rd-degree burn, often it's not very painful. And why is that? Well, if you totally sear off these nerve endings, how are these neurons going to even detect that change or what they would normally detect to begin with? So that's why 3rd-degree burns result in not as much pain as a 2nd-degree burn. Okay, so what we have here is a free nerve ending. So here is the end of an axon and all these small dendrites. So these dendrites, they actually don't have a covering. So think of it like someone who is naked. So when you're naked, you're exposed, right? And when you're exposed to your environment, you're more sensitive to your environment. So these free nerve endings are very sensitive. And just like when you don't have clothes, you're more sensitive to changes in temperature. When you have no clothes, you're going to be more sensitive to things that brush up against you and touch you. And same with pain. Like, say, which would you rather have, armor on or be completely naked if someone throws a ball at you and it hits you? So free nerve endings, they're important in detecting all these 3 general senses. And what we have here is a root hair plexus. So this is actually an axon, or actually dendrite. I shouldn't say This is a dendrite. Yeah, these are dendrites. Sorry, I know. Yeah, so these are dendrites. Bad physiology. Okay, all right, so these are dendrites because they're going to carry all the sensations towards the cell body. So this is a root hair plexus, and these are mechanoreceptor neurons. So what they do is wrap around each hair follicle, and so basically if you move this hair follicle, it's going to stretch those mechanoreceptors and those mechanically gated ion channels on these neurons and cause them to depolarize. So back to our original question before the lecture, why shouldn't you cut your cat's whiskers? Well, you might think that, oh, it's too long. But the thing is that your cat needs it. Your cat needs the whiskers because how are they going to feed? The thing about that is that their cats are actually able to use the root hair plexus or plexi, the four plexuses. So they actually are able to get that in sensations from their hair movements and actually feel around and use it in their sense of position and navigation throughout the dark. So, or if another example is like, say you're sitting, have you ever sat next to someone and all of a sudden they move, but you notice that they kind of brush up against your arm even though they're not actually touching your arm? But you can kind of tell when, or say like a bug lands on you and maybe not only do you have like receptors in your skin, but they can also distort the receptors in your root hair plexus as well. So even though ours aren't as sensitive as a cat, we do have sensation in our roots. Or heck, what happens when you pull out one of your hairs? Pretty painful, right? Why? Because you're distorting all those mechanoreceptors and firing and depolarizing those neurons as well. So tactile discs are interesting. They're what they detect. I like to think of them as like little keys on the typewriter or type keyboard. So these are also called Merkel cells. So I think a lot of times, I think the OpenStax version tries to move away from structures that are named after people, but I would just know both just in case. But again, at this level, I tend to give you both. Like I tend to, if there is what we call eponym, I tend to give you both. The non-named version, the version that's not named after someone, and the version that is named after someone. So I like to think of it that way. So these tactile discs, basically, they're like little pressure plates. What they do is like they detect some sort of things like, okay, where's the position of this thing, and notice that they're actually in right here. So you have to go through several layers of skin or of strata to actually touch these. So they're kind of there just to detect these general senses. Now tactile or Meissner's corpuscles, so they're actually kind of surrounded by this very cottony, jelly-like and loose connective tissue over here. So I like to think of these like I think when I look at this picture, I think of it like with those dandelions. You only need a slight touch, a slight breeze to set them off and have this like kind of scatter all these seeds. So this is the way I like to think about tactile corpuscles, like little dandelions. You only need a light touch. So this is what they detect. Just like these little dandelions, they only need a light touch to scatter the seeds. Same with these tactile Meissner corpuscles. And lamellae Pexinian corpuscles. So what they have is a bunch of layers and layers and layers and layers of collagen. And notice that they're also in the dermis compared to something like the epidermis. So these lamellae corpuscles are relatively deep, but they're also surrounded by layers of tough tissue. Versus a free nerve ending, which has almost no covering at all. So I like to think of it this way. Who's going to feel it more? If you punch this guy in the chest, or if you punch this guy who's wearing hundreds of shirts in the chest? The guy who is more naked, right, and exposed to the environment is probably going to feel it more than the guy who you're trying to punch through multiple shirts and layers of armor. So I like to think of this, and this is also related to the function of Pexinian corpuscles. So common mnemonic, Pacinian or Paxinian, they detect pressure, but think of it this way. Who's going to feel the pressure more? Someone who's wearing a lot of armor and a lot of layers versus someone who's almost got no layers. So that's the way I remember that these lamellae corpuscles, they detect pressure. And also that they're very deep as well. So you have to actually press really hard to get to that deep one. I only use that because I looked at the 100 shirt challenge. Alright, so Ruffini corpuscles. So this one is like interesting because they detect vibration. So here we have the dendrites over here, and they're going to carry this over here. And here is a dog. And what sound does a dog make? It makes this sound, right? So ruff, ruff. And what happens if you give a doggy a little, or if you've had a dog, what happens if you give them like a rope toy? What do they tend to do? They tend to do this, right? So you go, right? They tend to ruff, ruff, and they tend to like vibrate that rope back and forth. So this is the way I remember it. These bulbous corpuscles or Ruffini corpuscles, they kind of shake, so they detect some sort of shaking. They detect this vibration. So that's the way I remember that part. Okay, moving on. So now let's talk. We talked about how we're able to detect different types of changes in stimuli. Now let's talk about how these nerves are arranged. So what we have here is a peripheral nerve, and it's very similar to the skeletal muscle we talked about. Notice that we have all these like long fibers, and they're bundled into things called fascicles, and they're bundled into bundles of bundles. And this is what we're seeing with the scanning electron microscope. We notice that we have all these little bundles, some big, some small, grouped together, and these are what our peripheral nerves are made out of, bundles of bundles and bundles. They also have connective tissue that help to keep these bundles together. So here we have an axon from one nerve cell, and then we have the group of these axons bundled together into something we call a fascicle. And with this, so what we have over here surrounding each connective tissue between each axon right here is what we call an endoneurium. And then we have the perineurium around one fascicle. And then around each individual nerve, we have something called the epineurium. And epi, peri, and endo, where have we heard that before? Hey, sounds like our skeletal muscle organization, right? So bundles of bundles and bundles. So we have endomysium surrounding each muscle fiber, and then we have our perimysium surrounding each muscle fascicle, and then we have our epimysium surrounding each skeletal muscle. So this is just like, so you can use the same prefix, but so instead of we have endo being the most innermost layer of surrounding each myofiber, and then we have a perimysium, and then we have an epimysium in the skeletal muscle. But in peripheral nerves, what do we have? So peripheral nerves have fascicles, which are groups of nerve fibers, which are those axons. So around each nerve fiber, we have an endoneurium. Instead of an endomysium, we have a neurium. And then we have a perineurium surrounding each nerve fascicle. And then we also have an epineurium surrounding each peripheral nerve. Now here we have, so that's the way it's like. If you already know your epi, peri, and endo, you already got the levels of organizations in the nervous system or in the peripheral nervous system. Now this is the Martini version, and do I expect you to know this level of detail? Not really, but if you're going to 300 level, you have to know this stuff. But what I'm showing is that you have all these signals coming into something we call like various parts of the body. We have from the back, the limbs, and we have this whole network of all these sensory inputs and sensory neuron fibers leading towards the central nervous system. And one structure we see are things called ganglia. So ganglia are actually groups of cell bodies. And again, think about our neuron cell anatomy. You have that long axons, you have those dendrites from the cell body, but the cell body is the thickest part of a neuron, right? So it makes sense that if you have a bunch of the thickest part of a neurons gathered together, it would produce some sort of thick swelling, and this is what a ganglion is. So in the central nervous system, we call them ganglia, or sometimes also nucleus. I think they're actually trying to go more toward nucleus, and again, this is different from our cell nucleus, different from our atomic nucleus. So CNS is like, we're talking about the central nervous system and nuclei, we're talking about some group of neuron cell bodies in the central nervous system. Whereas in the peripheral nervous system, it's pretty consistent. Most textbooks and sources say a collection of cell bodies in the peripheral nervous system is a ganglion. Now, what we have here is that the ganglion is a group of so review here. But the interesting thing is that we also have these little groups, subgroups of ganglia. So we have somatic and visceral nuclei. Now, these are both sensory nuclei, so they both detect, these are cell bodies that detect something. But, let's see, actually, this is a little different. So or I'll cover this later, or should I cover it now? Okay, wait. Rewind. So these are sensory nuclei. So all these sensory signals coming in are going to end up at this part right here. And for the most part, and this rule kind of applies very in general. There are exceptions, but at this level, just know the general rule that applies to most cases. So most of our sensory sensations actually end up at the dorsal, the posterior part of our spinal cord. And so these incoming signals end up here, and they end up at these nuclei that help to interpret these sensory signals. So this leads to another common mnemonic. Another variation is same Dave, but what I learned from the previous professor who taught this several years ago is that she condensed it even more simple. So she has SAD MEV. So basically it's telling you that where do you find these nuclei, where do these nerves enter the spinal cord and central nervous system? So sensory stimuli tend to enter the dorsal area. So this is why it's called, the S is related to the D. Now this is another term. If you look at the organization of the central nervous system, think all the way back to when we talked about the divisions. So A before E. So afferent, or I'll mispronounce it and say afferent, because afferent signals are traveling towards the central nervous system. So again, sensation is carried toward your central nervous system, so sensory are afferent signals. And these tend to be located on the dorsal side of your nervous system. Now, motor nuclei, these carry out commands from your central nervous system to the rest of the body. So the corresponding part of this mnemonic is MEV. So MEV, like, it's a made-up word, but what's saying is like, okay, with motor commands, such as the ones that go to your motor neurons, that contract your skeletal muscles, these are efferent, the efferent signals. So that what they're doing is carrying out these effects. So motor and these efferent signals, they tend to be ventral. So they tend to be located on the anterior side of your nervous system. I know, so, but key points is like sensation is not the same as motor commands. And afferent, A before E, so afferent is carrying signals toward your central nervous system. Efferent signals are carrying commands from your central nervous system to other parts of your body. And they tend to be located dorsal and ventral respectively. Alright, so what we have here are these neurons. So we have dorsal root plus a ventral root, and they end up at something called spinal nerve. So again, dorsal tends to be sensory signals, and our motor commands nerves tend to be ventral, but they actually fuse together, and this is what we call a spinal nerve. And spinal nerves, for the most part, are what we call mixed nerves. So you don't have like a separate pathway just for your sensations and your motor commands. So most of your peripheral nerves are mixed nerves, meaning that they both carry sensation and motor signals. So they both can receive sensation and carry out commands. So except for the C1, and I put a little disclaimer here because I think there was another study that are like, we did find sensory neurons in the C1 peripheral nerve, but the C1 is predominantly, in terms of, if you had to like compare its proportion of motor nerves versus sensory nerves, it's vast majority are motor nerves. And then we're going to talk about dermatomes. So dermatomes, it looks like this really ugly leggings right here, but what are dermatomes? They're regions of the body that detect that detect sensations. And what we notice is that some of these dermatomes, these regions, are labeled with certain letters and numbers. And things like C3 or T12 or L5, S4, these kind of sound like vertebrae, right? And they do correspond to regions of the spinal cord that correspond to vertebrae. But it's not I'll talk about when we talk about the central nervous system, I'll explain it a little more. But yeah, so we have 8 cervical spinal nerves, 12 thoracic, and 5 lumbar spinal nerves. So it's almost exactly like our vertebrae, but there's extra cervical spinal nerve. And there's 5 sacral. So remember that the sacrum, even though it's one bone, it's from fused vertebrae. So with those little holes along the lateral sides of the sacrum, this is why we have those foramina, these little holes. They allow these sacral spinal nerves to exit from the spinal cord. And you actually have a coccygeal nerve that right here. So a small pair of coccygeal nerves. So even though it's like, and it's not just like 8 total. What we have is like actually 8 pairs up here in the cervix, 12 pairs, 5 pairs in lumbar, 5 pairs in sacral, and 1 pair in the coccygeal nerves. Now back to these dermatomes. It's a little confusing because they're showing you both the anterior and posterior at the same time. So it kind of looks like a Willy Wonka sort of thing, but I'm just going to reflect this. Now it looks a little more symmetrical. So here we're seeing the anterior, here we're seeing the posterior. So we're seeing that these sensations or these areas of the body, they carry sensations from these parts of the body to the corresponding spinal nerve. And this is the OpenStax version. So the OpenStax version only has the anterior part. And if I recall correctly, they actually don't bring up this figure until, chapter 16. So I'm like, where is this? I want this in chapter 13 when we're talking about peripheral nervous system. So this is why I think the OpenStax version is a little weird, because most other textbooks, they talk about this early on, but they decide to put it all the way to the let's ignore dermatomes until we talk about the neurological exam. A little bit frustrating. So what are dermatomes and why do we care about them? Well, dermatomes again are actually a part of the skin, or that's where we get the derma part from. But it's a part of the skin that's supplied by one single spinal nerve. So here we have the spinal cord, and here we have the T4 spinal nerve. So the spinal nerve is going to, at least the sensory part, is going to receive signals from this area around the same level as the nipple. And you actually have a paraspinal nerve, so you have a left and right. So this spinal nerve over here is going to detect on the right side of the body, and this spinal nerve is going to detect the left side of the body. So you have paraspinal nerves on either side of the spinal cord. Now with dermatomes, say someone touches here in this dermatome, well, what's that going to do is it's going to depolarize these dendrites and these nerve endings, and it's going to send signals along these spinal nerves toward the spinal cord, and this goes up to the brain, and these signals go up there, and then your brain detects, hey, something's touching me on that side of the body. So this is how this is very generally how you're able to detect sensations. So this is why we have somatosensation tests. So what we do is test things along dermatomes to see if there's any sort of, it's kind of like an indirect way of determining, or should I say indirect? It's a way of detecting whether there's some sort of spinal nerve damage. So what we do is try to elicit these different ways of somatosensation. And one common way is like just taking an object and applying it to that dermatome and seeing if the patient can detect it properly. So here we have the T1 dermatome, and notice that extends all the way across the chest and down the anterior of the arm. And what happens is so here we have C5 and T2, but this dermatome is going to run right along this path over here. And again, we have a paraspinal nerve, so it's going to have a left and a right. Now, say we apply a cotton ball to this patient's right arm. If their nerves are intact and functioning correctly, the signals will travel up this spinal nerve and towards their central nervous system, and their central nervous system will detect this sensation and say like, hey, I feel something touching me on the right side of my body. Now, what happens, say this guy likes to live dangerously and he gets into a knife fight, and the knife cuts over here and then it severs this nerve over here. What's going to happen to his somatosensation over here? Well, if he has a cut here and it damages nerve, what happens is that nerve endings, if they're not connected to anything, then they get degraded and they can't carry their signal. So say we test him after his knife fight and test him with that cotton ball in the same area. Well, since his nerve is damaged over here, he can't get signals from here past this where he got that cut earlier. So we can put that cotton ball here, but he'll be like, I don't feel anything. But how do we know whether it's a general nerve? Maybe he has other things going on, like it's not just that area. Maybe he has an inability to detect cotton balls altogether for whatever reason. One way you can double check your work and say like, hey, this guy might have something along his T1 is to test the other corresponding spinal nerve and dermatome. So we're going to take another cotton ball, put it on his left side of the body, and see if that same dermatome elicits the same response. And he's like, wait, I feel this one this time. So we're testing the same part of the body, but just on the opposite side. So he's able to detect it in the intact nerve, but this will make us think like, okay, one side is detecting that stimuli, but the other isn't. So the part that's not detecting it correctly might have some sort of nerve damage. So we're like, oh, he got into that fight, but may have damaged that nerve over there. Okay, so I think I'll cover proprioception next time because we're over time. But yeah, sorry if things are a little rocky for the next few lectures because I also had to reshuffle all my slides and the way I teach this part to correspond with the OSU. So it's going to be a little, at least until we get to the autonomic nervous system and special senses, there's a little bit of figuring out to do, but I'll try to do my best to kind of like make it a more logical, like how the OSU version of the textbook is compared to the OpenStax version. Again, it's not like I just want to throw a curveball, but I think the OSU version makes more sense overall. All right. Okay, thank you everyone for hanging on, and thank you to our moderators, and I'll see you all on Wednesday.